Welcome, everybody, to our lecture today on Gregor Mendel. Today we're going to talk about inheritance and how we get our traits from our parents. And we are going to talk about Gregor Mendel because he is kind of what we call the father of genetics. So before Gregor Mendel came around, there was kind of a general concept of blending, meaning that offspring would kind of be a mix of their parents, which makes sense. But as we learned more about genetics, we realized that doesn't really happen. What happens is you have these different alleles or traits, and depending on what is dominant and what is recessive, then that is how you get your looks or your hair color or your eye color. So it's a little bit different than just a simple blend. Okay? And Mendel was the first to discover this. And when he did his experiments, he came up with two different laws. One is the law of segregation, and one is the law of independent assortment. Okay? So basically, he said that inheritance involves a reshuffling of genes from generation to generation. He was an Austrian monk, and he had studied science and mathematics at the University of Vienna. He was a very bright man, and he worked with garden peas, little pea plants. And part of the reason he did that is because they were simple to work with, and they allowed him to be able to understand these laws of heredity and what was actually going on. And I mean, this was in the 1860s. So you have to remember, it's hard to remember, but imagine a time when there weren't microscopes or computers, or we didn't even know about chromosomes or cells. So how would you determine it? Traits of a plant. It kind of blows your mind if you stop for a minute and really think about what he accomplished in the time that he was doing it. Okay? So, Gregor Mendel, really amazing. So yeah, he used the garden pea. And he did this for several reasons. Basically, it was easy to grow. It had a short generation time, meaning that he could then get in there and kind of breed these peas together and then see what, the offspring looked like. So because of the short generation rate, it allowed him to be able to conduct these experiments rather quickly and get a lot of different results. Okay? So even though they do self-pollinate, they could be cross-pollinated by hand, meaning he was able to pick and choose which plants he wanted to breed and then try to get certain offspring, right? So it was, I'm sure, many trial and errors, but all these things allowed him to use the pea plant and come up with some really good results. Okay? So what he did was he did all these experiments where he literally took a green pea and bred it with a yellow pea, or a wrinkled pea and a round pea, and a short stalk with a long stalk, and all these different traits to try to determine what was going on. Okay? So the term true breeding means that it is homozygous, meaning it has the same traits, the same alleles. Okay? We're going to go through a lot of terms here, but we'll go slow and hopefully you'll get them all. So if you are trying to determine one trait, you would use a monohybrid cross, because mono means one. If you're trying to determine two, you use what's called a dihybrid cross. A couple terms or letters, I guess, to be aware of is P always stands for the parental generation. The F1 is the first generation of offspring, and the F2 is the second generation offspring. Okay, and then we have what's called the law of segregation. And that is the law that shows us that parents, their alleles are split, and one can go one way and one can go the other. And you're going to see that in an actual cross. So the law of segregation means that each individual has a pair of factors, or what we call alleles, for each trait. And these alleles can separate and go to either the sperm or the egg, depending on the plant. And then what happens is the gamete only contains one factor or one allele from each pair. And you basically get an allele from dad and mom, or male and female, depending on what's being crossed. Okay? So, each trait in the pea plant is controlled by these two alleles, which are basically just alternate forms of a gene. A dominant allele is denoted with a capital letter, and it will override or mask the expression of a recessive allele, which is denoted with a lowercase letter. And depending on how these are paired up, you can either be homozygous or heterozygous. So if you have two dominant alleles, two capital A's, let's say, you are homozygous dominant. If you have two lowercase a's, you are homozygous recessive. And if you have a big A, little a, different alleles, you are heterozygous. Not recessive or dominant, because you have one of each. You are just heterozygous. So, here are some things. Your genotype is the actual alleles that you carry. So maybe the big A. But your phenotype is how those alleles are expressed. Meaning, maybe you're tall. Okay, so your genotype is the letters, the AA, and the phenotype is the trait that is being expressed due to those alleles. So here is some of the things that he studied. He studied stem length, pod shape, seed color, flower position, flower color, and pod color. And it shows you right there everyone that's dominant and everything that's recessive. Pretty cool. So this is what I kind of just explained. But when you have two alleles of the specific trait and they are the same, you are homozygous. And that is your genotype. And if you have two alleles that are different, your genotype is heterozygous. And the phenotype is always the physical appearance that you are displaying because of your genotype. So phenotype is dependent on genotype. A dihybrid cross is when you cross two things, meaning you could be looking at color, seed color, and seed round versus wrinkled. Okay, so it's just a bigger cross. It's the same process. The traits are still independently assorting, but you're now looking at two traits versus just one, which is where we get the dihybrid. Okay, your P, your F1, your F2, exactly the same. None of the concepts change. You're just doing a bigger cross because, again, you are looking for two traits instead of just one. Okay, and we use what's called a Punnett square to do that. So the Punnett square is where you put all the alleles of, let's say on this picture, it's showing the eggs, the female on the top, and the sperm going down. So what happens is all these traits through independent assortment align, and then they come together, one from, traits from the eggs, traits from the sperm, and that is your offspring. So in this example that we have here, the P generation, the first boxes are capital T, capital T, capital G, capital G. So that means that that individual is homozygous dominant, and that means no matter how these alleles come together, it is always going to basically be a capital T, capital G. Okay, because that's all it has to give. The other parent there has little t, little t, little g, little g. So that means every time they produce an offspring or an egg or a sperm, it's going to contain a little t, little g. That's all they have to give. So in the F1 generation, every time those eggs and sperms come together, you're going to get the same mix. You're going to get a capital T, capital G, little g. That's all you can ever get. But now if you crossed two of those F1s with each other, you would get your F2 generation, and that's going to be a little different. So now you have to come up with all the different possibilities of those alleles, and you could get a big T, big T, big G, big T, little g, little t, big G, or little t, little g for both parents because it's the same plant. And then you see all those combinations, those 16 squares, and that is called a Punnett square. When you do a dihybrid cross, you always have 16 squares. You have 4 eggs and 4 sperm. When you do a monohybrid cross, you only have 4 squares because you're only looking at one trait. Maybe you're just looking at T's. So you would have 2 eggs on the top, 2 sperm going down. So you would only have 4. And you would only have 1 letter in those top and side boxes. You wouldn't have TG, you'd have just a T, whether it's a capital or lowercase. These are Punnett squares. You need to be able to do a Punnett square. If I give you a female and a male plant and I ask you to do either a monohybrid or a dihybrid cross, you have to be able to do that. So, a Punnett square is a listing of all possible genotypes resulting from a cross. So you have all the sperm genotypes lined up on one side, the egg genotypes are lined up on the other side, and the zygote genotypes, basically the offspring, are all placed within the squares, which is what we just looked at. Now, the more you do these Punnett squares, the more you become familiar with the probabilities. So there are certain probabilities of genotypes and phenotypes. So basically, you always get, depending on if you always start out with a homozygous dominant plant, like big T, and a homozygous recessive plant, little t, you will always get 100% of the offspring being heterozygous, because they have to get a big T from the egg, let's say, and a little t from the sperm. So every offspring is going to be big T, little t, or heterozygous. And the more that you do these, the more that you will become familiar with the values and the probabilities. Okay? So you just have to practice a few to understand how these probabilities play out and how to put, which allele in which box. So there are things called test crosses, and this has to do with individuals with recessive phenotypes always have the homozygous recessive genotype. However, individuals with dominant phenotypes have intermediate genotypes, meaning that they could be homozygous dominant or heterozygous. So a test cross can determine the genotype of an individual having the dominant phenotype. So test crosses are done to try to determine what the genotype is of an individual. Okay? And that brings us to the end of our first Mendel lecture. Please take some time, go over the readings in your book, maybe watch this again, look at all the posted videos, and make sure that you understand what a Punnett square is and how to perform both a monohybrid cross and a dihybrid cross. Please reach out if you have any questions or concerns. Have a wonderful day.